Hello, everyone. Welcome to the computer science seminar, which is also joined with the MIF++ seminar today in parallel on Zoom. It's a pleasure to introduce Professor Alex Hernandez-Garcia from the University of Montreal and also from the Mila Institute, collaborating with your Avengers group in Montreal, Canada. So Alex will talk today about generative modeling and active learning for scientific discoveries. Over to you, Alex, please.
Thank you so much, Vitaly. Thanks, everybody. I'm happy to see a bunch of people, even after I've heard a lot of people are on there, away from Liverpool. Thanks for coming over, and thanks for people online for attending as well. My name is Alex. I'm coming from Montreal. Well, I'm originally from Spain, but based in Montreal, and I'm very happy to spend some days here in Liverpool. I'm looking forward to talking a little bit about my research and discussing with you if you have questions. I'm going to talk about scientific discoveries and how we are using machine learning for these kind of problems, using both generative models and active learning on a blend of these two things. So the reason I work on scientific discoveries is, I started working on this out of an interest in applying machine learning for tackling climate change. Hopefully, you don't need to be convinced that this is an important topic. I'm going to try to move this down. Climate is warming. We're having a lot of trouble with this. There's a lot of things that can be done, but one thing is science, and one thing in science in particular is scientific discoveries. This is an illustration of how climate change is having a lot of impact on health, which is the primary reason why we actually care about climate change. But then why discoveries and why this is connected to climate change? Well, one thing is materials discovery. If you read the last assessment report by the IPCC, this consortium of climate scientists and other scientists who use this report every several years, they actually talk about the potential of new materials, especially in energy sector, to improve materials efficiency, energy efficiency, and so on. And there's a lot of promise on this, but it's a challenging problem. So it's not the kind of thing that one works a little bit on discovering new materials and every day finds a new one. And it seems that there's potential for machine learning and computer science also to make a contribution on this. And this is what I will try to convince you of throughout the presentation. I'm going to skip this part of motivation to get into the core of it. This is just a summary slides about this program that we're trying to carry out in our group. So we work on machine learning for science, in particular material discovery, and we also work on drug discovery. Both things are very much related, especially when we put machine learning in the middle. It turns out that it's quite convenient, because the methods that we are using for material discovery are actually used for drug discovery. And then also from a broader lens, like I've briefly mentioned before, these two problems in health, problems in climate are also connected. So that's why we work on both things. And something I really like is that there is a nice synergy that between the different sciences. So when you work on material discovery or on drug discovery with machine learning, this presents you a bunch of challenges that end up motivating new research on machine learning. And I find this very stimulating and exciting, also being a machine learning scientist myself. So the outline of the presentation is going to be an introduction about how generative modeling and active learning can be used for facilitating scientific discoveries in general, some starting from more of an abstract way. And then I talk about, I'll give an introduction to a relatively new method, not so new anymore, called generative flow networks or GFlowNets, and how this is connected to the challenges that appear in the use of machine learning for scientific discoveries. And then I'll talk about multi-fidelity active learning to bring all these things together. And I have for those of you who got the link of the slides, if you're interested, I left a bunch of slides about some particular applications, material discovery, and molecular conformations. I don't think I'll have time to talk about that during the presentation, but if there is interest, perhaps during the discussion, I can show some quick examples from the slides, and that's why I decided to keep it on the slide deck. But I'm going to focus on the first part. All right, let's get to it. Generative and active learning for scientific discoveries, and I think it's worth starting with the traditional discovery cycle. How traditionally scientists have been discovering new things. And I think this is an oversimplification, of course. It's just a schematic of how this can work. So usually we have scientists who accumulate a lot of knowledge and expertise over the years by looking at a lot of data, by learning a lot of stuff, and are able to come up with some ideas, hypotheses, experiments, things to try. Like that's what we mean here by the queries. And then usually there's some way to test these ideas or hypotheses in the lab with computer simulations. There's usually a way to get results, no? So this is what we mean by oracle in an abstract general way here. And the oracle gives us back some results, which we can conceptualize as data. Especially if I look at this problem through a machine learning lens, this can be conceptualized as data, information, you name it. And this is what informs scientists who are able to come up with refined ideas, refined hypotheses, right? So this is the traditional cycle. It has worked very, very well in a lot of problems. It relies on highly specialized expertise, and it can be time consuming and financially and computationally expensive, depending on the application. So if we want to do machine learning here, what can we do? Well, we have called this part data here, so probably an obvious thing to do is to train a machine learning model with this data and use the machine learning model to try our experiments instead of using the expensive, time-consuming oracle. So we can try our hypothesis on the machine learning model and predict stuff. Depending on the application, it's going to be one thing or another, predict stuff from the model. Of course, the model can be inaccurate. So it's going to be like problems associated to the inaccuracy of the machine learning model, but we can still use it. And chances are it's going to be better than random if we do things more or less. All right. I want to make a point here, which is that even if we have a perfect machine learning model, and even if it's like super fast, in certain problems, it's not enough. There are infinitely many conceivable materials, combinatorially many molecules. So we need something more. It's not enough to have a perfect surrogate model of our problem, even if it's super fast, to tackle these kind of combinatorially complex problems, because we cannot exhaustively search the entire space. We need some sophisticated methods to go about it and to explore these spaces. This is the case in material discovery and in molecular modeling and so on. So, for example, the usual number that is given for drug discovery is that there are like 10 to the 60 drug-like molecules. So this, I mean, it's hard to wrap our head around these kind of numbers. But if you do a back of the envelope calculation, even if you are able to screen like 1 trillion molecules per second, which is nothing we can do, it would still be like 10 to the 40 years to screen the 10 to the 60. So it's just impossible. We need a better way to decide which ones are we going to screen. to try. Okay, so that's why we introduce something else, another machine learning model, which is this generative model in the loop. And this generative model is what could serve this purpose of having a more of a sophisticated and smart exploration of the large space. And then altogether is bound through this active learning loop. So this is what when I say generative modeling and active learning together, I mean this schematic where a generative model explores the space, adjusts candidates, we try them with a machine learning model, we get predictions, and sometimes we verify our predictions with an oracle and repeat, acquire new data, and so on. And this is a way to explore and get better in our predictions and exploration. And yeah, I'll be arguing that this is a more efficient way to come up with new discoveries in science. But what are the actual challenges beyond what I've just said about the large spaces? So let me illustrate these challenges with this kind of toy Tetris problem that we're all familiar with. So imagine, very simple, I give you the task. I want to find arrangements of pieces of Tetris on this tiny board. And the reward, the objective function we're trying to optimize here is simply minimizing the empty space on the board. So very quickly, you can come up with a solution, probably. Maybe this one, if I can move this thingy. Let's see. So yeah, the score is the number of cells occupied by the pieces. So, okay, score 12, you find it very quickly in no time. I can make this, I can make this a little more challenging if I tell you, look, one is good to find one solution, but actually we need many solutions because in science this actually happens, that you find one solution to your objective function, but then down the downstream in the application, then it turns out to not work for some reason. In drug discovery, you know, there's all sort of things that matter, like toxicity of the molecules of the compounds and how well it is manufacturable and so on. And I'm happy to talk about this, but having diversity in the solutions is actually very, very important. So I tell you, look, I need all the solutions for this problem. Maybe you need a few more seconds to figure them out, but quickly you'll be able to find all the solutions for this small problem, which are these five objects, arrangements of Tetris pieces. Okay, easy enough. What if we make the board bigger and we have many more pieces to put on the board? Well, this stops being an easy problem. Now it's a pretty hard problem, I would argue. Still intuitive because we know what to do. We know what the task is. We need to put the pieces on the board, and we know the rules, and we know the objective function is to minimize the empty space. It's very clear, very well defined. We're able to get it. Again, we can find a bunch of solutions. If I ask you, you have to find all solutions to this problem, like I don't think you'll be able to do it. I don't think there's an easy algorithm for finding all this. There are combinatorially many. We need a lot of compute to do like a grid search about this. But this still intuitive. We can make it less intuitive, and it becomes an incredibly hard problem if now we have an objective function which we don't know what it is. So for example, I could tell you, I have done a search, and so far what I know is that these are good. This arrangements, these objects here, are near optima of the objective function. And then you have to start the search, and all you can do is query your objective function f of x and explore. So this is a challenging problem, right? Especially if the objective function is expensive and you cannot do a lot of queries on the function. And I would argue that materials and drug discovery share a lot of these challenges. So for example, if you want to design proteins as sequences of amino acids, if you have 100 amino acids, then you have a space of 10 to the 134 proteins. That's humongous. Pretty hard, even if it's just DNA sequences that are just for nucleobases, still a hard problem. And conceptually, it's much simpler than than the Tetris thing that we have just discussed. Very similar for molecules. I've mentioned before this number of 10 to the 60 drug-like molecules. This is just the subset of molecules that happen to share properties with known drugs. This is not even the number of potential conceivable compounds. This is just the ones that look like known drug compounds. We can represent molecules as strings or as 2D topological graphs. And we could see how some of the challenges I've been talking about would appear in this problem. So to summarize the challenges, first, I'm repeating this a lot, very large search spaces and also high dimensional. Like it's not just a couple of continuous numbers, it's highly structured objects. So this is the other challenge I'm talking about. Usually these problems entail searching structured spaces. So all these methods that work pretty nicely for low dimensional continuous spaces, because we know how to do optimization there, don't really work out of the box for this kind of problem. And that's another challenge. We need to come up with generative models or samplers that work as well for structured data. And that's not obvious. And then the aspect of diversity that I've mentioned. Most often we are dealing with objective functions or metrics that are underspecified. We have access to some objective function, can be a property of a molecule, a property of a material, something that's important for a problem, but by no means is that the only thing that matters in the program. And this is what we mean by underspecification. And this really happens across the board in most scientific discoveries that I encounter. So there are different ways of dealing with it, but an obvious one is saying, well, let's find diverse solutions, and this is something we can tackle, we can design for, and deal with this challenge. So to summarize, we want to discover diverse, high-scoring candidates in very large structure spaces. How to do that? So there are a bunch of available methods. For example, there is reinforcement learning, very good at optimization in complex spaces. So high dimensionality, structured objects, that's fine for reinforcement learning, but it tends to lack diversity. The standard reinforcement learning is very good at optimization, it tends to lack diversity. So not perfect for all the challenges. This is traditional sampling methods like MCMC, very good at diversity, less good at structure large spaces because of the problem of mode mixing. So we know that theoretically it can fit any function, but in exponential time. So not great for large structure spaces. If we take modern generative models in machine learning, we have, for example, diffusion, which is very good at learning from big data sets at scale. So this is the kind of models that are used for generating images and also used in different sciences. It has a lot of nice properties, especially works very well in continuous spaces. But I would argue that one of the drawbacks of this kind of diffusion models is that you have to really tweak it to make it work well with structured data. I'm not saying it's not possible; it is possible. You have to tweak it and find some tricks to make it work. So one method that was introduced in summer 2021, so a few years ago already, at Mila and Joshua Benja's group, was this generative flow networks. And it was motivated precisely by these challenges in sciences, in scientific discoveries. And it tries to overcome all these challenges in one go. So essentially, it can be seen as a method that performs sampling as sequential decision making. So it's not that it's a mix of MCMC and reinforcement learning, but it tries to bring the the best of both worlds. And it has actually connections to diffusion as well. I won't talk about that today, but it sort of sits in the middle of reinforcement learning, MCMC, and diffusion somehow. So what I'm going to do in the next few minutes is give a brief introduction to GFlowNets. So those of you who haven't learned about this have at least a high-level idea of this method, and then I'll talk about how it is used in active learning. So when I talk about GFlowNets, I like to mention three key ingredients that I find relevant and important for overcoming these challenges. The first one is that diversity is really part of the objective. It's not a byproduct that you can achieve. For example, in reinforcement learning, you can adjust you can add some regularization terms. So for example, maximum entropy regularization, and then you get diversity. Actually, there's a clear link between GFlowNets, which is reinforcement learning, and reinforcement, between GFlowNets and reinforcement learning through max entropy search. But in GFlowNets, diversity is part of the objective. This is what it is designed for. The way it is done is essentially because you have a reward function, and while reinforcement learning would optimize the reward function, GFlowNets learn to sample from it. And if you sample from the reward, then you're going to find, kind of by construction, you're going to find diverse solutions, unless you have a delta reward function. The second ingredient is compositionality in the generation. So instead of generating objects directly on the sample space, objects are generated sequentially by decomposing the sampling space into intermediate states, and this provides a decomposition into patterns, into generalizable patterns, and this is what can be used for generalization. So if X is decomposed into a bunch of states, intermediate states, it yields patterns that then can be picked by the deep neural networks that we use in machine learning, and that by now we know how to train. We know that they generalize, of course, to some extent, but they are very good at pattern recognition. So essentially, we can use a machine learning model that learns the transition function between two states. And if there are indeed patterns that are generalizable, this can be picked up by the deep neural network. So these are the three ingredients that I see as fundamental for in the GFlowNet framework. I'm just going to say a few more words about each of them. So first, about diversity and the idea of sampling from the reward function. So essentially, this is pretty much like an energy-based model. So we have a reward. We have a partition function Z that is, of course, the sum of the rewards in the sampling space. And we can define the likelihood of generating an X as the reward divided by the partition function. It's pretty much like an energy-based model. So if we are able to learn this likelihood, then we're going to sample proportionally to the reward, even if we don't know what Z is. So Z, of course, in a small problem like the one I used at the beginning, you can calculate it because you can enumerate the entire sample space, and you can calculate Z easily. But if you are sampling molecules, you will never be able to compute the value of the partition function for the entire molecular space. But it doesn't matter. You can still sample proportionate to the reward, even if you don't know the partition function. This is just to illustrate. So if we have this 2D problem, so one dimension, another dimension, we have this reward function that has high reward on the four corners of this 2D space. The idea here is that if there is structure in the reward function, so it's highly symmetric, then the promise is that perhaps a neural network trained in this framework might be able to figure out that there has to be some high reward this missing corner, this corner that we haven't explored yet, and kind of jump directly to it, which would be some kind of generalization. This is what MCMC, for example, would struggle with, especially if the modes are far apart. So yeah, something that is a bit different from most generative models is that P of X is modeled via a reward function and not through data, as it is done, for example, in diffusion models. So it's learned with the reward. So a few words about compositionality. So of course, compositionality is it's a well-studied aspect of linguistics, semantics, mathematics, and so on, and it's known to be a cornerstone of human reasoning. So it's known to provide the means for generalization. So if we take the Tetris problem before, instead of sampling directly on the sample space, we can decompose the generation of these objects into steps. So for example, start from the empty board, and add one piece at a time, essentially. So add another piece, eventually you decide to stop, get a score, and this, of course, would yield a tree of options. It's a tree because it's a very small problem, but in general, this can be a directed acyclic graph. So there can be multiple paths to each object, and this is something that GFlowNet nicely allows for, as opposed to standard reinforcement learning. And this is actually what gives the generalization. So this is what I mean by compositionality and by decomposing the generation of objects into steps. So this decomposition is what would allow learning complex distributions by breaking it down into multiple steps. So a bit more formally, you can see that you are decomposing, or you are using these notions from reinforcement learning of an action space and state space. So you can define transitions between states that are the actions, etc. And what GFlowNet learns to do is sample these trajectories of states. Okay, so finally, how does deep learning come into the problem? And I say deep learning, it could be shallow learning as well, but in general, we use deep neural networks for this. So like I said before, in general, you have multiple actions that, it wasn't the case in the previous case, but multiple actions that can take you to a given state. So consider this state ST, you can reach it from these two parents with different actions. And from it, there are also multiple children depending on the actions that you can take. In this case, the actions are, adding pieces at certain locations. And you can also stop instead of adding pieces, and then you would get your reward. So in the different framework, what the model is trained to do is to learn the value function. I don't want to use the value function because that has a particular meaning in RL. But it learns the value of the transition from ST to the next state ST plus 1. So that's what is parameterized by a neural network with parameters theta. And then informally, there's a theorem that tells us that if the flows, this is the flow function. If the sum of the flows that go into a state ST is equal to the sum of the flows that come out of that state, then you achieve this goal of sampling proportionally to the reward. So this is the main theorem in the original paper. So in the end, the problem boils down to figuring out, okay, how can we actually train this flow function parametered by a deep neural network such that it achieves this goal of minimizing the error in the flows. And this, I think, a slide about it. So this would be more formally this flow preservation condition. So you have a state S, and you sum the flow over the parents, and it has to be equal to the sum over the flow. Sorry, the sum of the flows over the children of the state S. and this is kind of like the statement of the theorem, that if that's true, you learn a probability of X that is proportional to the reward. Any question about that? Might be too fast or too. So we take this equation here, we can turn it into a loss function simply by moving one thing to the other side, taking the log and squared, and that's it. That becomes a loss function that we can minimize. So if we minimize, well, at convergence, if this loss function is 0 for all states, this is the flow matching objective that I've mentioned before, then we are guaranteed to sample proportionally to the reward, which is what we wanted at the beginning. And then, yeah.
So the final aim is to sample so that the loss is exactly 0.
The loss is like a way that has been found to achieve the goal of sampling proportionally to the reward. So this is a loss that guarantees that if the loss is 0, and then you start following the forward policy, so you start following this flow function that takes you from one state to the next. If the loss is 0, you're going to sample proportionally to the reward.
What should we do after that with all the samples? If we lay a first goal after that,
so
if all samples are ideal.
So if we have this state-rich problem, and the reward is the number of cells that are occupied by pieces, and we sample proportionally to the reward, then what we get is that boards that are fully occupied are going to be sampled more likely than others. And then you can, of course, tweak this reward function. You can take the exponential of it so that you give even higher probability to full boards compared to half full boards. So what you end up is sampling with high probability the good candidates, the candidates that have good scores. And this is the.
So in this case, you will be finding full solutions.
Full solutions that are actually different because if you train reinforcement learning on the same problem, reinforcement learning would be very good at finding more or less quickly one solution that minimizes or that maximizes the reward. If that's what you want, finding one, do reinforcement learning, reward maximization, you're going to be good, going to be very efficient. If you actually want to get a range of diverse solutions, ideally all of them, which is something that is often the case in, if you want to find molecules with a certain property, you want to find a bunch of them, then this kind of objective would be better suited for the problem because it would not be focused on just one solution. So there's a difference between mode seeking solutions and more like mode coverage solutions. So different is more mode coverage.
For molecules, so what kind of functions or rewards you're using?
So that it depends on the application. So if, for example, we're using GFlowNets for antibiotics discovery, and in antibiotics, one thing that is very important is the antimicrobial activity against a pathogen. So imagine we have a neural network that has been trained to predict the antimicrobial activity of a molecule, of a new molecule. We make that neural network the reward function. It gives higher reward if the compound is predicted to have higher activity against, for example, E. coli bacterium. Then if the GFlowNet is well trained, it's going to generate molecules that are diverse, or it's with high probability, let's say. With high probability, it's going to generate molecules that are diverse and are also expected to have high activity, antimicrobial activity.
Computed by this?
By the neural network. So yeah, if the neural network is really bad, then you're gonna get like really bad molecules. So you're gonna be limited by that reward function that you use. If you are tackling a problem where the reward function is fully reliable, then you're good. That's what you want.
That is in this example.
Like in this example is perfect, because it's easy to compute. So you would sample perfectly from the reward. Yeah, and I put this trajectory balance objective, so I won't get into the details, but there are, apart from this flow matching objective, that was the initial objective in the first paper. There have been other have been proposed with different objectives have been proposed, all having the same property that eventually you sample proportion to the reward. And this one turns out to be more efficient to train, because while this one is local, it talks about a transition from state S or from S prime to S. This one is about full trajectories. So the credit assignment is more efficient. If you're interested, let me know and we can talk about the details after. I just want to accelerate to get to the active learning part. So I have a couple of slides just to show quickly that this has been extended to multi-objective GFlowNets. If you have multiple rewards that you want to satisfy at the same time, then you can do it as well. Everything, of course, gets a little bit more messy, more complicated, but it's doable. So far, I've been talking about discrete problems, but we also extended it to the continuous spaces and hybrid, so you can mix discrete hybrid objects and things still work. So this is just an example of a continuous space where you have a high reward in the corners, and this is how GFlowNet learns to eventually sample with high probability on the four corners of the 2D space. And yeah, I put this slide here in case you're interested to read this review paper where we talk about pretty much like what I'm trying to do with my talk. I talk about the benefits of this approach for scientific discoveries. If you're into code, I would like to give it a try. There is this open source library that we have in a bunch of papers, and I think by now it's easy to extend and to apply to different problems. So if you ever want to give it a try, don't hesitate to reach out. I'll be happy to help. Any questions about this intro to GFlowNets? Does it make sense? The high-level idea of this? I've skipped a lot of details, of course, especially the mathematical details and the proofs and so on, which I'm happy to talk about later, but hopefully the idea is there. So I would like to spend the last 10 minutes or so talking about active learning and how using GFlowNets actually in this active learning problems brings benefits precisely because of the diversity aspect. I have a few slides talking about related work. I think I'll go quickly through that, but who is familiar with Bayesian optimization? Yeah. So what we're going to be doing is pretty much Bayesian optimization with a twist, which Bayesian optimization does optimization of an objective function. And what we are saying, so this is Bayesian optimization, what we are saying is in certain problems, especially in scientific discoveries, optimization it falls short at achieving the goals because it's too kind of narrow focused on just okay, what is the optimum of this objective function that I have? And it turns out that this function, the objective function, will not capture everything we need. So that's why we try to sample from the objective function instead of just optimizing. But otherwise, it's gonna be very similar to Bayesian optimization. We just don't call it Bayesian optimization because it's not optimization, but it shares a lot of the properties of it. There is something called active search. We shared some properties, but I'm going to skip. There's a family of algorithms called quality diversity. Is anybody in the room familiar with quality diversity algorithms? Okay, sadly, because I keep trying to find like words on this. You can teach me. It's a different family of methods, very different to difference. They are based on genetic algorithms. But I put them here because actually they have the same objectives. They want to find solutions that have quality, so good scores, and that are also diverse. The approach is very different, but the goals are very similar. And then active learning is a term that is very general, and it essentially keeps the idea of, the main idea there is that, well, there's some sort of active loop in which we acquire new data and repeat and repeat. So it's hard to boil down like a clear definition of this because people use it in different ways. But I use this term active learning to mean that. So Bayesian optimization for me would be like a type of active learning. But of course, there's a lot more to it. And in particular, I'm going to talk about multi-fidelity active learning, which is the situation in which we not only have one oracle, which is what I showed at the beginning, we have actually multiple of them. The reason is that if we have just one oracle, we're missing out in what science offers. Because usually the best oracle we can have is very expensive, very time consuming, has a lot of limitations. So for example, it can be, I mentioned before the problem of antibiotics, finding a new antibiotics. So we can do experiments in the lab to measure the antimicrobial activity of a molecule, but that of course costs money and it takes time because you have to synthesize the molecule that has its own challenges, and then you have to do an experiment, grow bacteria, bacteria culture, and get some results. So it's going to be hard to even do just optimization. Even if you just want to do optimization, it's going to be hard to do that because of the challenges it entails. And to simplify things, we can assign a monetary value to this, a cost, and we can say, I have it in Canadian dollars here, sorry. We can say £1,000 each evaluation. So imagine the Tetris board. It costs £1,000 to evaluate one Tetris board. What can we do? Well, if we have cheaper ways to evaluate how good a candidate solution is, then we could potentially take advantage of that and accelerate the exploration. So maybe we have the very expensive oracle, but then we also have a slightly inaccurate oracle that costs us £100. And we even have a noisier oracle that is just £1. Maybe we have some that are even cheaper. And we can say, well, but this is very inaccurate. Well, but inaccurate is useful if it is not random. So one thing is random, another thing is inaccurate. So there is information theory would tell us that if it is not random, there is information and we can do something with it. But the question is, how can we actually integrate multiple oracles in this problem? And it turns out that there is a little bit of a literature on multi-fidelity Bayesian optimization, but there was very, very little, if I'm not saying like no literature at all, on this problem of integrating multiple oracles when the task is actually sampling and having diverse solutions, especially in domains like molecular modeling, material discovery, and so on. And that's why we propose this active learning algorithm that I'm going to explain in a second. So again, the goal is twofold. We want not only high-scoring candidates, but we want them to be diverse, so same as before. It's not enough to have just one good solution. We want many, and ideally different. What we did here is to evaluate it on a biological sequence design problem, so DNA, peptides, or proteins, and small molecules. That's what I will show in the results part. But let me walk you through the algorithm. So in the single fidelity version, you have one oracle. You have if you have some data, you have a small data set, you can annotate your set of X's on your oracle at M, and then you get a labeled data set that then you can use to train a surrogate model. Like in Bayesian optimization, if you're familiar with Bayesian optimization, that's it. And you have a probabilistic surrogate that models the posterior distribution of the oracle, given a new sample X and the data that you have, D. What we do differently than Bayesian optimization is that we introduce a GFlowNet, and if we have an acquisition function that tells us the worth of annotating a new candidate, Bayesian optimization would optimize the acquisition function. With a GFlowNet, what we do is sample. We want to sample from the acquisition function so that we get a diverse set of candidates. So in the end, we can sample a batch and select the best of them, the top B, and then annotate them again with our oracle and repeat. And this would be an active learning loop in which we are constructing a diverse batch of candidates, annotate them, repeat. Now I told you that we want to make this multi-fidelity. Multi-fidelity is the case where we have a set of oracles. We're going to refer to them like F1, F2, up to F capital M. And we just assume lots of generality. This is going to be like more inaccurate and less. So F1 is going to be the cheapest and also the most inaccurate. The cost, we are going to refer to it by lambda. So lambda is going to be the cost of annotating one candidate with the oracle. And now, everything is the same, but just a little bit more complicated because everything has to be more, everything has to include the multi-fidelity aspect. So now we don't have only X's, we have X's and M's. So the M is going to refer to the oracle that we use to annotate each candidate. So we're going to have a batch of candidates. Each candidate is going to go to a different oracle potentially. And then we end up with this triplets of candidates, the value of the oracle, and the index of the oracle that we use to annotate that candidate. So you see, it's not too hard. You see that it can be quite messy. Now we need a surrogate model that also needs to be multi-fidelity. Just a little detail. See that we don't have multiple surrogates. We have one surrogate that is multi-fidelity. So it takes as input both X and the index of the oracle. And this gives us, it models the posterior distribution of the oracle given X, M, and the data. Of course, the acquisition function also needs to be multi-fidelity. We take one from the multi-fidelity Bayesian optimization literature. That, of course, inversely weights the value of the acquisition by the cost. And then the additional twist we introduce here is that GFlowNet, because it's very flexible model, we can make it sample whatever we like. We make it sample not only the candidate X, but also the oracle. So after it is trained, it's able to decide, this is something like this, this is a good candidate, but for this oracle. And this is a good candidate, but for this other oracle. So that's the idea of the goal in mind. Everything else is the same, sample a bunch of candidates that are hopefully diverse, and then it gets distributed to the different oracles, and then repeat. So this we evaluated in a bunch of problems, and it worked quite well. We need to convince you, but I'm running out of time. So I'm just gonna quickly show you one result here. And then you can believe me or not that it worked for the other problems. You can check it out. So the first baseline, or let me say first what we're showing here. So I have this plot where the x-axis is the fraction of the total budget that we use in an experiment. We're gonna do multiple rounds of data acquisition, and when we reach one, we have exhausted the available budget. And on the y-axis, I'm showing the mean score, in this case, an energy, of the top 100. This is to kind of show that we want a bunch of them to be good. So to have this idea of diversity and good scores. And this problem in particular was for generation of small molecules where the property was the adiabatic electron affinity, which is relevant in organic semiconductors. With a single fidelity model, so also with the GFlowNets, but single fidelity, this is the curve that we get. So indeed, as we get more data, we get better mean scores. If we do the same, but we select random candidates, still ranked by the acquisition function, which is what makes it good. So that's a strong baseline. It's slightly worse than single fidelity. In some problems, random candidates ranked by the acquisition function is a very strong baseline, so it could be better than that. In this case, it was. This orange line is already a multi-fidelity GFlowNets active learning thing, except that the fidelities were random. So we didn't let the GFlowNet choose, and we instead assigned the random fidelity. And that already improves the performance over the single fidelity, indicating that actually having access to multiple oracles is probably a good thing to do. And then finally, if we let the GFlowNet select the oracle, it improves even farther from the previous baseline. So indicating that it is more efficient than the exploration of these kind of molecules in this case. We did other experiments with similar results, but I'm not going to stop to the details. So I'm going to stop there. And so this is peptides, and this is a small toy case. So I'm going to stop there and take your questions if you have any. And I have other slides with more details, but I think I've shown you the most important bits of the algorithm and the main results. So let's finish acknowledging Nikita, who was a student who did a lot of work in this program, and Moksha, Chenkao, and Yusha worked on these projects. Yeah, thank you.
Thank you, Alex. Stop the recording.